Okay, this is part 4. This slide is, you've already seen this slide, where I talked about how we're not going to cover the organelles. Instead, what we're going to focus on are the 3 components of a muscle fiber or a muscle cell, the myofibrils, the SR, and the T-tubules. So we've covered the myofibrils. That was talking about the myofilaments, thick and thin, actin and myosin. Actin has these other regulatory proteins, troponin and tropomyosin. So our focus now is going to be on the SR and the T-tubules. So the SR, or sarcoplasmic reticulum, is just smooth ER in a muscle cell. And it surrounds each myofibril. And I'm going to talk about this in just a second. Paired terminal cisternae. But basically, we know from the previous one that calcium gets released. Calcium binds to troponin. Troponin moves tropomyosin out of the way. And then tropomyosin, once that's moved, myosin head can bind to the active site on actin. So then where does this calcium come from? The calcium comes from the SR. We're going to see a particular part of this SR where it's more concentrated. That more concentrated area of SR is called the paired terminal cisternae. So that's what we're going to see here. Right here and right here. On either side of this yellow line, which is a T-tubule, on either side of that, those are the paired terminal cisternae. You still see SR here, but it's just a lot more concentrated here. And think about it. Right here in this H zone, we don't have thick and thin overlapping, so there isn't as great of a need for calcium there, right? Because there isn't any actin. If there isn't any actin, there's no troponin for that calcium to bind to. So these, the 2 blue, though, those are the paired terminal cisternae. So they will release calcium. Calcium will bind to troponin, move tropomyosin out of the way. Myosin head can bind to the active site on actin. Contraction occurs. You're going to want to go through that a couple of times. Now we're going to continue our reverse engineering. What tells the paired terminal cisternae and the SR as a whole to release that calcium? And it is an action potential. So an action potential is the signal that tells the paired terminal cisternae to release the calcium. We're going to go over action potential in great detail, but for right now, all you need to know is that it is carried along the sarcolemma. It's carried along the plasma membrane. And so what we're going to see, these T-tubules, are these deep invaginations. Remember what we said? We said that every single muscle fiber has to be hit with a nerve, and then each muscle fiber, right? Here's the sarcolemma. So this is inside a muscle cell. We're going to need an action potential to hit this myofibril, this one, this one, this one, and this one. So what we're going to have is we're going to have invaginations of the sarcolemma that go deep inside. It's going to enhance communication inside the muscle fiber. So those T-tubules are just invaginations of the sarcolemma. So the action potential, you guys know I can't draw. Here's the sarcolemma. Here's this T-tubule. The action potential will travel along that membrane and then go down the T-tubules. When it goes down the T-tubules, it tells the paired terminal cisternae to release calcium. And this cluster of the paired terminal cisternae and the T-tubule is called a triad. And so extensions of the sarcolemma that penetrate into the cell's interior at each A-band, I-band junction, that and the terminal cisternae form triads, conduct impulses to the deepest regions, and those impulses, those action potentials, cause the release of calcium from the paired terminal cisternae. So now we know we have an action potential that travels along the sarcolemma, down the T-tubules, tells the paired terminal cisternae to release calcium. Calcium binds to troponin, moves tropomyosin out of the way. Myosin head can bind to the active site on actin. Muscle contraction occurs. And so the last thing that I want you to know is that when contraction occurs, the thin filaments slide past the thick ones. Right? So what's going to happen is that H zone is going to get smaller. So they overlap only slightly.